Assalamu alaikum dear student. In today's class we will talk about the phylogenetic tree and we will discuss about different part of phylogenetic tree and we will discuss how to interpret phylogenetic tree. So this lecture is very important for those students who are involved in research and also for the bachelor student who are always facing problem to describe a phylogenetic tree in their exam. So in today's lecture we will discuss what is a phylogenetic tree, the type of phylogenetic tree, and how we can construct a phylogenetic tree, and how to describe, how to interpret the result of a phylogenetic tree. So I hope this lecture will help you in your exam as well as in your research. So first of all, we will start from the phylogeny. That what is phylogeny? So the evolutionary history of a species or group of related species. In phylogeny, we studied the evolutionary history of a species or a group of species or related species. And the discipline in which we study these, it is called systematics. And systematics, what we do, we classify organisms and determine their evolutionary relationship. Now, what is a phylogenetic tree? A phylogenetic tree, it is also called evolutionary tree. Okay, the phylogenetic tree also called evolutionary tree, and it is actually the graphical representation of the evolutionary history of biological sequences and allow us to visualize the evolutionary relationship between them. So phylogenetic tree, it is the graphical representation of the an evolutionary history of biological sequences. Okay, what it will help us, it will help us to visualize the evolutionary changes between species, the evolutionary changes between organisms, the divergence between organisms. A phylogenetic tree represents a hypothesis about evolutionary relationship. So phylogenetic tree will always present a hypothesis about what? About the evolutionary relationship between species, among species, and about some specific type of organisms. If we are discussing phylogenetic trees, so certainly there are many types of phylogenetic trees. So the first one is cladogram. So in this type of phylogenetic tree, what we can conclude from phylogenetic tree, and what are the differences between the cladogram and other type of trees. So in cladogram, it display only branching pattern. In cladogram, it will show only the branching system of evolutionary relationship of organism. Okay, so cladogram, it is unscaled. Okay, it is unscaled. The branch length do not reflect the amount of evolutionary divergence between taxa. Two things important here. The first one is the it will give you only a branching pattern of the species. Okay, the second important thing, it is unscaled and it will not provide any information about the evolution and about the divergence of different taxa. Okay, so in cladogram we can only make a phylogenetic tree which show only the branching pattern. Now, on the other hand, what is phylogram? In phylogram, it represents the evolutionary relationship among organisms, which is similar to cladogram. But by showing both branching pattern and the amount of evolutionary divergence. In phylogram, we can know about the evolutionary divergence of species. In cladogram, we cannot do this. We cannot get information about the evolutionary divergence, but in phylogram, we can get information about the evolutionary divergence of species. Phylogram are scaled, which mean that the branching length are proportional to the amount of evolutionary divergence. It means here the branch length have meaning, and it show the evolutionary divergence. It show the amount of evolutionary divergence. So here you can see in these trees, the first one, this one is cladogram. So in this one, the cladogram here, you can see in cladogram, we have four taxon A, B, C, and D. But we don't have any scale. It only show a pattern of branching. Okay. But here in phylogram, we can see that it has the same taxon, but we have a bar here. We have a bar which show the divergence in million year ago. MYA mean million year ago. So it show that here you can see that the both are in the same position, both are in the same position, so it will not give any information about the evolution. But here you can see the A and B diverge early and C and D diverge later. Okay, so this one you can see here that it will give us information about the evolutionary divergence, that which species diverge before and which species diverge later. Okay, this is also the example of cladogram. Here you can see these are in the same line. Okay, the same line, it will not give you information about the evolution, and it will not give information about the time, the divergence time. But in phylogram you can see here this is different, this is different, these are different. Okay, and you can see here that if we make a scale here, so you can see that the divergence time of these taxon or these species are different from each other. These two are the same. Okay, the first one is cladogram with no such information. Okay, this one with no such information, but here you can see there is a difference in their length, the branch length here show divergence, it show time divergence, and it show the evolution. Now, we can make phylogenetic tree by different methods. As we discussed in our previous lecture, when we download the FASTA sequence, we blast the FASTA sequence. You can check my video for that how to make a phylogenetic tree by using FASTA sequence, and how to download different sequences from NCBI database for construction of phylogenetic tree. So in that video we explain and we use one method which is called neighbor joining method, but here we will discuss about the different methods used to construct a phylogenetic tree. Now, here the phylogenetic tree, there are two main type on the basis of which we can construct phylogenetic tree. The first one is distance based method. Okay, what we do in the distance based method, or what the software are doing during whenever we are using software for phylogenetic analysis. So what is the procedure or the mechanism of those software by calculating or constructing the phylogenetic tree. So calculating evolutionary distance between sequences by using substitution model, which are then used to construct a matrix. So in this one, in this type of tree, we use substitution model, and then from model we will construct our distance matrix. So and this distance matrix we can use to construct phylogenetic tree. So there are two main type of phylogenetic trees by using this method. The first one, unweighted peer group method with arithmetic mean, it is mostly called UPGMM method, and the second one is neighbor joining method. So these two method can can construct phylogenetic tree based on distance based method. It means they will use substitutional model. Okay, first they will use substitutional model, and then they will convert into distance matrix, and then they will use the distance matrix for phylogenetic tree. Okay, the second one is character based method. Okay, now what is the difference between distance based method and character based method? So as the name indicate, that the character based method involve analyzing sequence data, directly examining the sequence character rather than relying on pairwise distance comparison. So in this one, it will rely on each sequence during the analysis. It will not construct a distance matrix. It will include all the data of DNA or protein or any data provided to the software. So it will analyze every sequence one by one, and then it will make a phylogenetic tree on the basis of that one character, one nucleotide sequence. Okay, so these method evaluate all sequences at once by analyzing one character at site at a time. So okay, so this, it will not make a distance based matrix. It will analyze the sequence one by one, and it will construct a phylogenetic tree on the basis of all the sequence analyzed. So this, what is the advantage, and you can say the disadvantage of this? So this character based method, it is more accurate than distance based method. But one problem here, for example, if we want to do neighbor joining method, if we want to do distance based method, so if we want to construct a phylogenetic tree, so maybe the phylogenetic tree will construct in 5 minutes, 5 minutes. But if you upload the same data to any software, for example, mega software, and you want to construct the phylogenetic tree by maximum likelihood. This method, these two methods, maximum likelihood and maximum parsimony, these are character based method. If we use this software to construct the phylogenetic tree from the same data, maybe it will take 5 hours. So this is the point is here, that these methods are more computationally intensive and require more sophisticated statistical model. So it need more computationally intensive, like more powerful computers, more powerful resources, and also sophisticated statistical models. So sometimes it depends on our data, what we want to do, what we want to visualize by phylogenetic tree. We want to visualize the divergence, we want to to visualize the evolution, or we just want to identify the bacteria or identify the organism. So it's depend on our data, that what we want to visualize. So on the basis of your requirement, on the basis of your result, you can change your statistical your method by for the construction of phylogenetic tree. Now, a phylogenetic tree represents a hypothesis about evolutionary relationships. So we all know that the phylogenetic tree represents a hypothesis. How? Because it will give you an idea that this species evolved from this, and this character are common character between two species, and this character is like a separate character, and it's only belong to the ancestor species. So it is a hypothesis. It will give you a hypothesis. How? Because it depend because phylogenetic tree, it depend on many factors. It depends the species included, you included, how many species included, how many taxa included, how many information you included in the phylogenetic tree. So it depend, the result will depend on your provided data. Now we will discuss about different parts of phylogenetic tree. So for example, if we have this A and B, two taxon, okay, and here C and D. So if we have these, so you can see here, this is the branching point, okay, the branching point, and this is the sister taxa. So each branching point represent the divergence of two species. So this point, it will represent the divergence of A and B, okay. And here, these two species are called sister taxa, okay, which are diverged and which are emerged from these common ancestor here, okay. This is the common ancestor. Now a rooted tree, so we have two type of trees. For example, if we have a tree A, we have B, then we have C and D, okay. For example, if we have this tree and we have a root here, root, a common ancestor here, so we can say that this is a rooted tree. But if there is no root, if there is no root and we have only this, so it is unrooted tree. So a rooted tree include a branch to represent the last common ancestor of all taxa. So if we have a rooted tree, so this is the last common ancestor of all these taxa. Okay. And now what is a basal taxon? Sometime we have a tree and we have a taxon is at the base. Okay, for example, here we have E. Okay, so now this is the basal taxon. This is at the base of the tree, and this basal taxon, the characteristic of basal taxon, it diverge early. It diverge early in the history of a group and originate near the common ancestor of the group. So this is the common ancestor and it originate here. So it is diverge early from A, B, and C and D. Okay. Now, this is another phenomena. A polytomy is a branch from which more than two groups emerge. So we will discuss in the next slide that what is polytomy, okay, and how it's emerged from the from more than two group. You can see here. This is a typical phylogenetic tree. So if you see here, this A, B, C, D, E, F, G, these are called taxa. These are called taxa. One is taxon. Okay. This B and C, these are called sister taxa because they share the common ancestor. Okay. Now this A and B and C, these also share the common ancestor here. Okay. This G, it is called a basal taxon because it's diverged early and it's near to the ancestral lineage. Okay, so this the ancestral lineage, it's near it and it's diverged early from the other taxon. So that's why, and it is at the base of the tree, it is called a basal taxon. We usually select outgroup as a basal taxon. Okay, now this point, this is called branch point. Okay, and this point here you can see this branch point from a polytomy. So polytomy, we discussed that it is unresolved. It is a confusing situation because we don't know about the D, E, and F, which one is evolved early, which one is evolved later. So it, because they share the common point, so it is called this. In this one you can say that which more than two groups emerge, and here more than two groups, one group is this one, the other group is this one, so more than two group emerge from here, so it is called polytomy. Now this point, you can say this point represent the common ancestor up from A to G. Okay, this one represent the common ancestor from A to F. This one represent the common ancestor from A, B, and C. Okay, so I hope it is clear now. Now, if we discuss these, so this A, B, C, and D, these are descendant. These are descendant, and this is the common ancestor. And these are descendant from this common ancestor. And this axis, it show time or it show divergence. Now, where the species A, the species B, it share a character here, so we can say if two species diverge at this time, so we at this point, so we can say this is the speciation event. The speciation event, if you go in detail, so species formation occur here. So this is the ancestral lineage. If we come here, the A, B, and C, so this green color, this green color show unique, the B is unique, this blue color show C is unique. But here if you see to, if you come here, so this line, it share both B and C. So both B and C they share some character here. Okay, we will explain in detail. Here you can see A, B, and C. These are three taxon. So this one, this point, it is only this it is only correct. These character only unique to C. This point, it is common ancestor of both B and C, and this is the common ancestor of A, B, and C. So this is how to analyze a phylogenetic tree in different parts of phylogenetic tree. Now the question, what we can and we cannot learn from a phylogenetic tree, that what are the information that we can get from phylogenetic tree and what are the information that we cannot get from the phylogenetic tree. So the phylogenetic tree show pattern of descent. It always show a pattern that these species evolved from other species, but it will not give any information about the phenotypic similarity. We cannot get any information about the phenotypic of organisms. Phylogenetic tree do not indicate when species evolve and how much change occur in the lineage. It will not give you that information that when exactly this species evolve. If you want that you want to know that when the species evolve, then you need to put some information about some fossils records about your data. It should not be assumed that a taxon evolved from a taxon next to it. It doesn't mean. What does it mean? It doesn't mean that this A and B evolved from this A. So phylogenetic tree will not give you any information that this species evolved from a particular species. Now, what is cladistics? Cladistics is a term used in phylogenetic trees, in phylogenetic analysis, and systematics. So, cladistic is a biological approach to group organism by common characteristic. So, it will you can say classify organism on the basis of different or common characteristics, that some when there are some characters are common in A and B, so it will make a clade. Okay, if some character are common A, B, and C, so it will make another a separate clade. Okay, so what is a clade? A clade is a group of species that include an ancestor species and all its descendant. Okay, so the clade, that if you want this A and this is B. Okay, so the A and B will make a clade. Why? Because they share some character. They will share some character, maybe the character related to color, maybe the character related to height of the body. So it will make a clade because of some common characteristics. Now, a valid clade is monophyletic clade. So there are three type of clades, that monophyletic, paraphyletic, and polyphyletic. Now, the monophyletic clades, it consists of the ancestor species and all its descendants. Okay, so in this one, the monophyletic clades, you can see here, this is a monophyletic clades. Okay, A, B, and C, these it is called clade because it share the common ancestor here. Okay, so monophyletic signify it consists the ancestor species and all its descendants. So this is the ancestor species, this one, and A, B, and C are descendant. So this clade include all the descendant and the ancestor species. That's why it is called monophyletic clades. Now, if we come here, so D and E, this is a monophyletic clades, but F and G is another monophyletic clades, but G is remaining here. Okay, G is not included here. Okay, so all descendant are not included there. Okay, so a paraphyletic group consists of an ancestor species and some, but not all of the descendant. So in paraphyletic, in paraphyletic group, all the descendant are not included in the clade. So that's why it is called paraphyletic clade. In monophyletic clades, all the descendant includes. with ancestor species, but in paraphyletic clade, all the descendant are not included, but some are includes. Okay. Now, the polyphyletic clades you can see here, and polyphyletic grouping consists of various species with different ancestors. So now, how? Because you can see here that A, B, C, these are monophyletic clade and the common ancestor is here. But if you come here, D, E, F, and G, these are different and the common ancestor is here. So in polyphyletic, but we included this D here. Okay, so this is called polyphyletic clades because here you can see that various species with different ancestor. So with different ancestor you can include species with different ancestor. So this is called polyphyletic clade. So whenever you are describing your tree, whenever you are discussing your tree in some article, so you have to check whether your species is making monophyletic clades, whether your species will make paraphyletic clades, and whether your species is making polyphyletic clades. Here's another example. This one, the green one, it share a common ancestor here, so we can say it is clade. If we select this one, so we can also say a clade. Okay, but here if we come here, it is also single but a common ancestor, so we can say it is a clade. But here you can see that if we take this one, so it share a common ancestor, we can say it is a clade. But if we took this one, so the ancestor it doesn't share a common ancestor, so we cannot say it is a clade. And the same here, it is not share a common ancestor because this one is remaining, so we say it is not a clade. Okay. Now, we will discuss about the shear ancestral and shear derived characters. That what are the character which are shared with the ancestor and what are the character which is derived or which is only found in the descendant. Descendant and taxon, you can say these are the same. Okay. The character which are present in the descendant are derived character, and which character they are, those character which they share with the ancestor is called shear ancestor character. So in comparison with its ancestor, an organism has both shared and different characters. So an organism has both shared and different character with ancestor as compared to the ancestor. Now, what is a shared ancestor character? A shared ancestor character is a character that originated in an ancestor of the taxon. This character is originated in the ancestor of the taxon. It is not like it is not the derived character in the taxon. It is this character is present in the ancestor as well. Now, a shared derived character, it is an evolutionary novelty unique to a particular clade. So now this character, this shared derived character, this character is not shared with the ancestor. It is only present in the taxon or it is only present in the descendant. A character can be both ancestral and derived depending on the context. So it depends on the tree, it depends on the position that the character are shared with the ancestor or it is only a derived character. So now if we draw a phylogenetic tree using the derived character, how we can do that? When we entering or when we draw phylogenetic evolutionary relationship, it is useful to know which clade a shared derived character first appear. So you have to check it that this a particular character is derived in which species at what point. Okay, so you will understand from this. As we discussed earlier, that what is a clade. So a clade with clade, when two species A and B share some character, so it will be in the same clade. So clade with shared characters. Okay, now we can see here, you will understand here, we have six type of organisms here: the lancet, the lamprey, the base, which is a fish, the frog, the turtle, and leopard. Now we will discuss the ancestor character and the shared derived character. Okay. Now we will come to this table and we will make a table. On the x-axis we will put the organism names or the taxon or the descendant, or the and the y-axis we will put the characteristics. Okay, so first we will come to the first row. So vertebral column. So vertebral column, we will see that vertebral column, it is present in all descendant or in all organism or not. So we will see. So in Lancet we don't have Lancet doesn't have the vertebral column, so it will be 0. So in other all they have a shared characters. Okay, they have shared this character vertebral column. So you can see that this all have the vertebral column. Okay, so this is a clade vertebral column. So this is a clade, this is a derived character in this clade. Okay. Now, if we go to the jaws, so in jaws, lamprey also don't have the jaws. Okay, so this one you can say that these four, they have the share character of jaws. So they will make a separate clade on the basis of this derived character jaws. Okay, the third one is four walking legs. So now if when we come to the four walking legs, so it will exclude the fish, and these three, it will have share character only the four walking legs. Okay, then the fourth one is amnion is a membrane. You know, maybe the zoology student, they know about it, that it is a membrane found in mammals. So the membrane, these two they will share this membrane characters. Okay, and the hair, the leopard only have hairs. Okay, so it is the character which is only belong to this one. So if we come here, so you can see here the ancestor character. This is here what is the ancestor character? The ancestor character, that this is different, this one is different. Okay, now the ancestor, doesn't have this vertebral column, but the descendant have the vertebral column. So they will make a separate clade. You can see here, this is, you can say this is a clade. Okay, on the basis of vertebral column. Okay, but if you go to the jaw, so then this is the separate clade on the basis of jaw, and then it was classify a separate clade on the basis of leg, and then so on. So now the same we can do in the phylogenetic analysis and the DNA sequences, that they will see the character, they will check the character, that what are the common character between organisms. So they will make a clade on the basis of those characteristics. So these are the derived and shared characteristics. Now, what is outgroup? We always include an outgroup in our phylogenetic analysis, and why we use this outgroup. So a species, it can be one, it can be two, it can be three. It means you can include one species, you can include two species, you can include more than two species. So a species or a group of species that is closely related to n group. Now, what is n group? If a phylogenetic tree, for example, in this one, you can see here, you can see here that this one is an outgroup. Okay, and all these are n group. Okay, so these a species or a group of species that is closely related to n group, the various species being studied. Okay, so the outgroup will be definitely closely related to the n group. The outgroup is a group that has diverged before the in group. So whenever you want to include a phylogenetic, you want to include outgroup in your phylogenetic analysis, so it must be diverged before the in group. Okay, because then it will come to the base of the tree. So scientists dealing with the systematic and dealing with the phylogenetic analysis, that they always use outgroup which is related to the n group, and which have divergence history, or which have time divergence as early than the n group. So they compare each n group species with the outgroup to differentiate between the shared derived and shared ancestor character. So what they will, why they use the why they use this outgroup, because they want to check the derived character and the shared character, that what are the character the species or the descendant they share with the ancestor, or what are the character which the descendant derive or evolve during their life cycles. Now, the phylogenetic tree also show the branch length. We sometimes we are making phylogenetic trees, so we need to show the branch length. So what does it mean? In some tree, the length of the branch can reflect the number of genetic changes. So it will reflect the number of genetical changes that have taken place in a particular DNA. The branch, for example, if we have a phylogenetic tree A and B, okay, and then we have C, D, okay. So this the branch length. These length, it show the divergence. It show the evolution. It show the divergence among these species, and it show the divergence from the ancestor species. Okay, and it depend on the data you provided. So it can be like if you provide nucleotide data, if you provide protein data, and if you provide some fossils record, so then it will change the representation of the tree will change with the provided data. The other type of tree, we can include the chronological time, in the chronological time in which the tree shows that when a particular species evolved, when a particular species diverged from other species. Okay, so in this type of tree we always need fossil record, and there are different software. One software which is called beast, which is the best software to draw a phylogenetic tree, but we need fossils records of some certain taxon in that tree. We have to put the fossil record, and then we can draw a phylogenetic tree which can show the chronological time, and it also show that when a species evolved. So in the x-axis or in the basis, you can say that it will show the million years ago, that how many million years ago this species evolved. So you can see here that if we discuss this tree, so it means the Drosophila, it's evolved more than 542 million years ago, and then the Lancet evolved as 452 years ago, and then the zebrafish you can see here. So you can see that when maybe 300 years ago, and the frog when this maybe evolved as this one here, and then the human and mouse, and then you can say that the human in mouse, it's evolved or diverged in 65.5 million years ago. So this is it is a difficult complicated phylogenetic tree, but you always remember that what is the meaning of phylogenetic tree, and why I included in this in my study, in my thesis, in my research article, and what I want to show to the reviewers, what I want to show to the readers, what I want to show to what I want to display here. So it depends on you, that how you interpret your tree, how you interpret your result. But these are the basic information about a phylogenetic tree, and there are many softwares, many different algorithms, many different softwares by using which you can construct phylogenetic tree. But sometime when you want to construct a complicated phylogenetic tree. So then you need more data, more metadata to include in that experiment and those analysis, and you can draw a more complex and more like informating phylogenetic tree. So I hope this is enough for today, and I hope you will get some information from this lecture, and I hope you will remember these information whenever you are doing your research studies. And if you have any problem, if you have any like a difficulty to understand in some points, so you can write comments in the comment box, and you can contact me, so I will try to explain again if you have any point. Thank you very much.